All right, okay, so we'll move on to, in my unbiased opinion, the most exciting part of the day, nanomedicine. We have a very influential and gifted speaker from MGH. You don't like it? Okay. I have to be a little bit more moderate about her accomplishments, but she's very well known in the field of molecular imaging and nanomedicine. She's been working on siRNA delivery with nanoparticles that are also imageable. So she's really focusing on developing new cancer therapeutics, I believe, to use siRNA in conjunction with diagnostically active nanomaterials. Given her current state, maybe she should also start working on more on immunology and allergy treatments. So you'll hear her voice is a little bit affected by the spring, the season, and the induction of certain allergies. But I'm very excited that she's gonna. She had the opportunity to come here and to present the latest work she's been involved in. Anna Moore.
Thank you, Willem. Thank you, Zahi, for having me here. And I do apologize for my voice. Turned out that I'm allergic to New York or whatever is blooming here.
Allergic to us.
Everything's upregulated in my body, so I apologize for that. So I'm going to talk about image-guided precision nanomedicine for cancer therapy. And first, I'd like to say that the genetic application to medicine is really not something completely new. And the term pharmacogenetics was actually introduced more than 50 years ago. I believe it was 1959. But it's only now that the idea of using patients' biological information, genetic information, to improve and tailor treatment has gone from niche to mainstream. And the reasons are quite simple. First is the cost of sequencing a genome. We know that now it went down dramatically. It was $95,000 15 years ago. Now it's only $5,000, and probably next time I speak here, it's going to be 50 cents. But more importantly is that with the development of all these new techniques and next generation sequences, White House initiative came up last year in January of 2015, and they realized that they should start spending money in this direction. And they actually invested about over $200 million to precision medicine, directly to precision medicine initiative, with $70 million that went right to NIH. Anybody seen that money yet? So, the idea of precision medicine is to customize care based on genotype and phenotype of the patient. And this can be actually achieved if the patient's genetic code becomes part of your personalized medical record and directs personalized therapy. So the doctor actually can direct treatment based on that genetic information. So precision nanomedicine is something that I'm going to talk about. It's a part of precision medicine, and it's an emerging area of disease treatment and prevention that based on exploiting and exploring genetic variability of the patient. So once the genetic code of the patient is established and genomic variabilities are identified, treatment can be designed based on entirely rational, individualized, and targeted approach. And this approach is based on a modular nanoplatform that could be used for design of unique diagnostic or therapeutic agents that are, again, based on patient needs, and by needs, I mean variabilities in genetic code. So this nanoplatform would consist of the modular nanocarrier that would consist of, that would incorporate therapeutic, imaging, and targeting components. So in the vision that we're developing in my laboratory, The therapeutic component would consist of RNA-based molecules that would bind to target RNA molecule and ultimately cause its degradation or translational repression, something that we know as a phenomenon of gene silencing. So imaging component of this platform could help with monitoring the delivery and actually imaging the outcome in certain cases. So ultimately, we think that imaging can inform therapy and help us to build, to design more efficient and more personalized therapies, if you can say it that way. And ultimately, imaging can help to determine the cause if therapy fails in a given patient. So let's imagine a scenario when a patient is diagnosed with breast cancer. So the patient usually undergoes biopsy and a routine blood draw. And these biosamples are then analyzed using global gene expression analysis. This analysis would identify mutations and changes in genetic and epigenetic targets that are very specific to this particular patient malignancy. We can then design nanodrugs that would be uniquely tailored to these particular mutations and correct those mutations if necessary. So these nanodrugs would carry RNA-based molecules, and these molecules, again, would act on principle of complementarity. They would bind with target RNA molecule and cause its degradation. And all of that can be achieved under image guidance, which is, of course, provide us a lot of information as to where our drug goes and whether it's efficient or not. However, the day when we can really use the genome of an individual patient is not there yet. So what we need to do right now is basically start using the molecules and genetic mutations that we are aware of and target those molecules that we know causing cancer and causing mutations and causing metastatic spread. So, in other words, we can only use molecules that have been already identified. And in my presentation, I will show you how we approach this in my lab and how we actually apply this precision nanomedicine for cancer treatment. So since we're talking about RNAI, which is RNAI interference, just a few words, and I'm sorry for maybe most of you know what this is, but just in case. So RNA interference, it's an innate cellular mechanism for post-transcriptional regulation of gene expression. And the result of it is a gene silencing. So there are several ways of silencing genes. First of all, we can inhibit mRNA by small interfering RNA. Next, we can modify gene expression by inhibiting microRNA or by introducing microRNA mimic. In the next few slides, I'll show you exactly how it works. Therapeutic applications are multiple, from cancer to hematology to infectious diseases. It's used even in ophthalmology to treat macular degeneration. Of course, it's all at the level of clinical trials right now. But basically, looks like it's a very efficient way to treat disease in a flexible way as well. So how does RNA interference work? So this picture actually shows you the regular way how the cell makes a protein going through mRNA, DNA, mRNA, protein. We all know that scheme. Now, if we have genes that are implicated in cancer, they can be silenced, and they can be silenced at the level of mRNA. So how siRNA therapies work? They work by introduction of double-stranded RNA that's called further by the series of enzyme digestion. They produce small interfering RNAs, and they eventually they bind to mRNA and cause mRNA degradation, or as called translational repression. This mechanism is very precise, and you can actually silence just about any genes. Now, the other mechanism that I mentioned involves an endogenous RNAI mechanism, involves microRNAs. So microRNAs are non-coded double-stranded RNA molecules. They regulate gene expression, and their function is yet to be determined for every single pathology. But as of right now, there's about 5,600 mRNAs have been identified, and they actually regulate a huge part of human genome, up to 60%. So what happens is that these microRNA, they bind to mRNA, and they inhibit gene activity. So if microRNA binds to mRNA, so the protein is not synthesized. In many pathologies, including cancer, microRNAs are dysregulated. They're either upregulated, which causes binding to mRNA and inhibition of protein synthesis, or they downregulated. In this case, mRNA continue the synthesis of the protein, and in many cases it could be the cause of the disease. So the idea behind microRNA therapy is to normalize microRNA expression of the cells. So how do we do that? So if we have a situation, just like I said, if microRNA is downregulated, so we need to add microRNA mimic. Those are double-stranded RNA. They would, and the addition of those microRNA mimics would result in decreased protein expression because they would bind to mRNA. In case if microRNA is upregulated, we would need to add single-stranded microRNA inhibitors. They would bind to microRNA and leave mRNA free and able to produce proteins. So we have basically three pathways that we can exploit for therapeutic purposes. We can introduce siRNA to silence oncogene. We can introduce microRNA mimics, or we can introduce microRNA inhibitors. However, no matter what mechanism we use, we need to deliver those intact RNA molecules to the target tissues. And this is where we have significant problems. Well, first of all, RNA molecules are very unstable and they're degraded in serum. But we know that we need to deliver an intact molecule to the target site. RNA molecule, due to its charge, cannot simply cross cell membrane, but the interference mechanism happens in the cytoplasm, so that membrane crossing has to happen. Furthermore, free RNA cause off-target effects due to partial sequence complementarity, and also can activate innate immune system. So there are several ways to fight that, and in many laboratories, starting with the early 2000s, investigators suggested to use various delivery molecules. And this is just a small list, I guess, mentioning just about everything that has been tried, from lipid molecules to viral vectors, and of course various nanoparticles. But unfortunately, if you look at clinicaltrials.gov, the clinical trials that include siRNA molecules, you'll see that out of all of them, there's just really a handful of clinical trials that are ongoing, and virtually none that have been successful. There is actually only one that was partially successful, and this is a multi-center phase 1 clinical trial using lipid nanoparticle formulation that target VEGF and kinesin spindle protein that causes cell cycle arrest at mitosis. And these nanoparticles were used to treat liver metastasis, in endometrial cancer, and that was the only one that was, like I said, semi-successful. The reason I'm saying semi-successful because it did treat metastasis in the liver. This is a CT showing you that after 12 to 40 rounds of treatment, there was a disappearance of multiple hepatic lesions. But the problem with these particular nanoparticles is that they only go into the liver, and they only target liver. So if you have metastatic spread somewhere else or tumor somewhere else, you're not going to see any success. And in fact, this is another example of actually failed clinical trial. This is there was the first clinical trial that used nanoparticle as siRNA delivery system that was injected intravenously. It was a collaborative effort between Caltech, UCLA, MIT, and Calanda Therapeutics. And these nanoparticles consisted of cyclodextrins, and they were targeted to transferrin receptor that is upregulated on many tumors. And the siRNA was to M2 subunit of ribonucleotide reductase. This is an enzyme implicated in gemcitabine resistance. So this is just basically the mRNA mRNA transcript, downregulation of mRNA transcript after intravenous injection of these nanoparticles in 3 patients. And you can see that there was a significant downregulation of mRNA, but for some reason the protein level was still very high in the patient. And actually, further safety trials also failed, and it was a decision that was made not to advance to phase 2 due to safety concerns and lack of efficacy. And actually, intellectual property and licenses were returned to Caltech. So that was a total failure. I think, and many investigators after those unfortunate clinical trials thought that maybe the reason for that is that really we don't know where those nanoparticles are going. And maybe there is a way to know where they're going if we use imaging. So I'm not saying that imaging would resolve all the problems, but at least partially we would know where those nanoparticles are going. So in my laboratory, we're using magnetic nanoparticles as a flexible modular platform for nanodrugs, as we call them. I'm not going to describe it in details because you guys all know how to synthesize them, most likely. So iron oxide nanoparticles, coating, in our case it's dextran. We can make them targeted by attaching peptides, attaching antibodies, aptamers, you name it. We can also attach secondary imaging reporters, dyes or isotopes for correlative secondary modality and microscopy if necessary. And as a therapeutic moiety, we use oligonucleotides in the form of siRNA or single-stranded nucleotide if we want to target microRNAs. So over the years, we learned how to modify those nanoparticles, because most of you probably know in the early 90s, late 80s, early 90s, those nanoparticles were used as imaging agents, and this is how we started using them as well. And back then, we didn't even know about siRNA. We never even heard that word. And only when it turned out that the siRNA couldn't be delivered, we thought, okay, so these nanoparticles could actually serve as a delivery vehicles for those oligonucleotide molecules. But again, back then, we didn't know about it, and we only wanted to know whether those nanoparticles can even be taken up by the cells. And I just can't help but show this picture from actually almost 20 years ago, believe it or not. This is in vitro incubation of peritoneal macrophages and tumor cells with rhodamin-labeled iron oxide nanoparticles, showing that after incubation they go inside the cells and accumulate in tubular lysosomes. It's strange to me to look at this picture right now and to tell my students, can you imagine we couldn't make color pictures back then? We didn't have that capability, and they said, well, just leave it like this, it looks vintage. I said, okay, well, that's good. Over the years, we learned not only how to direct those nanoparticles to the cells in vitro, but how to do it in vivo. And we actually published a lot of papers on that, and one of them was on the cover of Cancer Research, one of the first targeted nanoparticles actually directed to tumors. So, and now again, the question was, can we use these nanoparticles as carriers for oligonucleotide delivery to tumors? And the second question was, can we use these nanoparticles for monitoring the delivery non-invasively? And one of the first study that we did is almost 10 years ago. We synthesized nanoparticles to test whether we can deliver small interfering RNA in a modular system. In a model system. So we synthesized nanoparticles consisting of iron oxide covered with dextran. We also introduced a secondary imaging modality. We modified them with optical dye, CY5.5 optical dye. We modified them also with myristylated polyarginine peptide to make them cross cell membrane and accumulate in the cytoplasm. And finally, we conjugated them with RNAI to GFP. And this is a tumor system that we used. We used an animal bilaterally injected with cells expressing GFP and RFP, red fluorescent protein. So we expected that these nanoparticles would accumulate in both tumors, but only in one tumor it would cause downregulation of GFP transcript and elimination of GFP signal. So first we showed by magnetic resonance imaging accumulation of these nanoparticles in the tumors. So this is a pre-contrast and 24-hour post-contrast image. And as we expected, we saw accumulation of these nanoparticles in both tumors, which was confirmed by near-infrared optical imaging because we had CY5.5 optical dye on these nanoparticles. So they accumulated in both tumors. And then we used fluorescence imaging to monitor downregulation of GFP transcript. So this is a white light image, just a photograph of the animal before the injection of the nanoparticle. And I should say that all injections were done intravenously. And this is 48 hours after the injection. You can see that the tumors grew a little bit. And now we see the same animal in the fluorescence channel, in the green channel and the red channel. Of course, we took separate images and then we just superimposed them so you can see both signals on the same image, RFP and GFP. And this is a color coding of these signals. And 48 hours later, we saw a virtual disappearance of GFP signal. And the RFP tumor was just as bright as before the injection, only it grew over the period of 48 hours. We performed RT-PCR and we showed that there was a significant downregulation of GFP transcript in the animals injected with these nanoparticles carrying RNA to GFP compared to animals that were injected with either saline control or with mismatch control nanoparticles carrying mismatched RNA. We also performed microscopy and we showed that there was a significant reduction or virtual elimination of the green signal in GFP expressing tumors compared to RFP expressing tumors. And blue here is the nanoparticles. So there was a first indication that we can deliver active RNA to tumors. And the next question was, can we deliver a therapeutic RNA and target a therapeutic target? So we synthesized nanoparticles that were similar to the previous ones. There were two differences. One is that we made them targeted, and I'm not talking about it here, but because it's a subject of a separate talk. But we made them targeted, targeting underglycosylated mucin 1 antigen. This is an antigen that is expressed on many tumors. Over 50% of adenocarcinomas express this antigen. And the second thing we did, we conjugated these nanoparticles to RNA targeting BRCA5 gene, and this is a gene that encodes for survivin. And survivin is an anti-apoptotic protein in tumors, upregulated in many tumors. So we thought that by targeting this gene, by downregulating survivin, we could cause the reduction in tumor size. And of course, we synthesized scrambled nanoparticles for control purposes. So we first showed the accumulation of these nanoparticles in breast cancer tumors. So this is in vivo MRI pre and post. These are T2 maps pre and post. Again, this is an optical imaging showing accumulation in the tumors of these animals. And finally, we performed a treatment. The therapy was delivered to the animals once a week for 2 weeks. So basically, there were only 2 injections. And those 2 injections caused a significant reduction in tumor size, shown here on this graph. And not only that, but it also caused a significant increase in apoptotic nuclei, as shown by the tunnel assay. And here is the quantitation of this assay. So in our studies that we continued based on this data, we decided to combine treatment of nanoparticles targeting survivin with conventional chemotherapy. But the idea here was that maybe we can reduce the dose of conventional therapy. And in our case, we used doxorubicin, which, as you all know, is a very toxic chemotherapeutic. So we used the combination of anti-survivin nanodrug with a low dose of doxorubicin, and you can see that there was a significant improvement in the survival of animals injected with this combination. We also worked around the combination, and we showed that there was actually sequence dependence here, which again I'm not focusing in right now because I don't have time. But that's a study that's ongoing, and we're trying it with various chemotherapeutics in various combinations. So in the last part of my talk, I want to tell you and share one very exciting study that is ongoing in my lab, and hopefully we will translate it into clinic in the near future. And this study deals with therapy for metastatic breast cancer based on silencing of microRNAs. So we actually focused on breast cancer because the statistics of breast cancer is really, really poor, metastatic breast cancer, that is. So we know that 90% of cancer death occurs due to metastatic spread and not primary tumor. In fact, women don't die from primary tumors these days because they are removed, and then after the course of chemotherapy and radiation therapy, if necessary, patients continue their healthy lifestyle. So if you look at the survival, so the survival at stage 0 is 100%. And stage 0 and 1 is 100%. But it's a different story when the cancer spread to local and distant sites, and survival drops to 22%. So in fact, the estimated cases for 2015 is over 231,000 it's in the United States, and 40,000 deaths in 2015. That's the estimated, but I'm afraid that these are the real numbers. So, so the reason we thought that our nano platform is very suitable for treatment of metastatic cancer, because there were several publications that came from Bob Weinberg's lab that identified microRNA 10B as actually primary driver of metastatic spread in breast cancer. They looked at microRNA 10B and showed that it was really upregulated in metastatic patients. It's overexpressed in metastasis versus primary tumors. It's also overexpressed in primary tumors of patients with metastasis compared to patients without metastasis. In other words, this microRNA is well validated in clinical studies in patient tissues. So our approach was that we thought that we could use our nano platform to inhibit or eliminate the formation or proliferation of lymph node metastasis. And there were actually two goals in mind. One is to prevent metastatic spread, and another one was to arrest the growth of already formed metastases. So based on our previous knowledge and previous knowledge of how our nanoparticles work and where they go, we synthesized nanoparticles that carried oligonucleotides targeting microRNA 10B. So we also added CY5.5 optical dye for correlative optical imaging and microscopy. In our in vitro studies, we showed that cells, breast cancer cells, MDA and B231 cells take up these nanoparticles, and this uptake downregulates microRNA 10B transcript very, very significantly. But most importantly, in vitro studies showed that treatment of the cells with nanoparticles carrying oligonucleotide to microRNA 10B abrogated migration and invasion of the cells. So if you look at these two pictures here, you can see that the cells that were treated with PBS, they continued to migrate into high serum concentration wells, and they continue to invade. The same thing you can see with the scrambled nanoparticles. They continue to invade and migrate. But in the case with the cells treated with our, as we call it, a nanodrug against microRNA 10B, the cells do not migrate and they do not invade. So these were very encouraging in vitro studies, and we went into in vivo studies. And first, we wanted to show the delivery of the nanodrug to to orthotopic breast tumor. So we used MDA and B231 expressing luciferase, human breast adenocarcinoma. First, we showed the delivery by MR. This is pre-injection of the nanoparticle. This is post-injection. And also we confirmed that by optical imaging. We showed that nanoparticles go to the primary tumor and also go to lymph nodes in these animals. So after we confirmed the delivery by two independent imaging methods, we went on to therapeutic studies. And we used this model, as I mentioned, MDA and B231. And those of you who work with this model knows that it's a very nice and very predictable breast cancer metastatic model. So the way it works is when you inject a primary tumor, in about 4 to 5 weeks, you see the formation of lymph node metastasis. And this is a very consistent process. Maybe this is why PerkinElmer charges $2,600 for the cell line. But it's worth every penny because it's really worked very nice. So, this in this case, we used this model after 2 weeks after implantation of the primary tumor, prior to formation of lymph node metastasis. And again, because these cells express luciferase, we could monitor and know when those metastases are formed. So we only took animals that had no formed metastasis. So those animals with primary tumors were treated with either nanodrug against microRNA 10B or scrambled nanodrug. And you can see that those animals didn't develop any metastasis, and the animals that were treated with scrambled nanodrug, they all developed lymph node metastasis, and this effect was highly reproducible. So the take-home message from these studies was that this nanodrug is capable of preventing lymph node metastasis in a breast cancer model. We've also performed histology. We showed that those lymph nodes of the animals treated with active nanodrug were clean with a normal lymph node architecture, whereas lymph nodes of the animals treated with scrambled nanodrug had characteristic appearance of lymph node metastasis. So the next question was, what happens if we already have metastasis? Can we stop metastatic spread in those animals? So we took animals about 4 weeks after implantation of the primary tumors with animals with already formed lymph node metastasis. And again, we knew that they had lymph node metastasis by bioluminescence imaging. So we treated those animals again with scrambled and with nanodrug against microRNA 10B, and we showed that there was a significant increase in metastatic burden in animals treated with scrambled nanodrug, and the metastatic spread was actually arrested in animals that were treated with nanodrug against microRNA 10B. And here is the graph that shows you the bioluminescence signal in metastatic lymph nodes of these animals. Interestingly, we also imaged a primary tumor, and we noticed that the primary tumor had no effect, the injection of these nanodrugs had no effect on the primary tumor. Primary tumor continued to grow in both animals. And this was an expected effect because this microRNA is only responsible for invasion and migration, but not for cell proliferation. We know that metastatic spread is actually a result of two events. One is repopulation of the cells from the primary tumor, and the second one is proliferation of the cells that already invaded the lymph nodes. So in order to eliminate the first root of cells, we surgically removed primary tumor and performed treatment regimen as I just described. In fact, this is what we would see in clinic because, like I said, primary tumors are normally removed. So, and we observed that there was a very similar picture. So we saw that there was a reduction, not reduction, but arrest at this point. It was the arrest of lymph node metastasis in animals treated with a nanodrug against microRNA 10B, and there was continued growth of metastatic spread in animals treated with a scrambled nanodrug. So it was a very interesting actually result because it looked like there was a difference in microRNA 10B biogenesis, whereas in primary tumor it was only responsible for migration and invasion. In lymph node metastasis, it also responsible for cell proliferation, something that we really didn't expect to see. But you can look at this figure actually, and so you can look at this figure actually, and you can see that we only treated those animals for 4 weeks, and you can see that this curve is actually going up, and you may say, wait a second, you really didn't arrest the growth of this metastasis. It's only arrested for a few weeks, and then it tried to go up again. So in our next study that was actually just came out in Cancer Research, we combined treatment of animals with a nanodrug against microRNA 10B and a low dose of doxorubicin. And you can see that so we had five groups of animals. One treated with PBS, another one treated with a scrambled nanodrug. The third group was treated with scrambled nanodrugs plus low-dose doxorubicin. The fourth one was treated with nanodrug against microRNA 10B. And finally, the fifth one was treated with nanodrug against microRNA 10B and low-dose dox. And in this, all these groups except the fifth one, you can see that the metastatic spread was stopped, at least in this group, and then it went up again. And all those animals eventually expired. But in the fifth group, we actually were able to eliminate metastasis only after 4 weeks of treatment. And the star here means that we stopped the treatment after 4 weeks. And all those animals went on to the end of their natural lifespan without treatment and without metastatic recurrence. So this is the bioluminescence signal from those animals. You can see that after the treatment was stopped, the bioluminescence signal in those animals continued to be under the baseline, and the survival in this group was 100% compared to other animals that were in other groups that finally they expired. So The important thing in any studies like this is also to evaluate toxicity of the nanodrug. And here we performed initial toxicity studies. We showed that actually the nanodrug in combination with low-dose dox doesn't really cause any significant toxicity in vivo. The blood chemistries were not affected significantly, and H&E histology showed no effects either. And actually, if you look at those animals, you can probably easily see which one is treated and which one is not. You can see that the treated animal actually is active, and the weight is significantly higher compared to this animal with metastasis that is limping and lost a lot of weight. And this is a still picture of those animals. So at this point, we're thinking about clinical translation of our studies. There are two possible scenarios that we're looking at. The first scenario, we think that women who underwent neoadjuvant therapy followed by surgery but still have a residual disease, in other words, they have incomplete response of the treatment, would benefit from this therapy, especially those who were diagnosed with triple-negative breast cancer, the most unfortunate type of breast cancer. And in the second scenario, that's probably the most likely scenario where we're going to start our clinical studies, is when patients have their primary tumors removed, but then they have their metastasis detected or recurrent, and this is where we begin the treatment. So this is where this study is right now. And again, like I said, we're looking for clinical translation, and hopefully it is successful. But the very last thing that I wanted to say is that what I just told you is our understanding how precision nanomedicine could work in the particular case of targeting microRNA 10B or targeting a survivin gene. But in general, if we talk about image-guided RNA-based precision nanomedicine, there are still some challenges and unresolved issues. And I think that this has a very high potential because of the entirely rational, individualized, and targeted approach. I think that nano platform that I presented is very unique in the terms that it gives us an opportunity to design all those diagnostic and therapeutic agents based on patients' needs, on patients' genetic profile. We have an added value, which is an image-guided delivery. But of course, we realize that the impact can be limited because of drug toxicity, of induced immune response, and a big issue of tumor heterogeneity that I'm not even touching right now, again, because it's a separate topic that we can discuss. Finally, there are some unresolved issues, like I said. One of them, the big one, is regulatory challenges, because we cannot really change, we cannot really go to FDA every single time asking for every single treatment for every single patient. So we have to think how to unify that process. And of course, we realize that generation of genetic information would create a big data that would need to be analyzed, interpreted, shared, and stored, which would create probably some privacy issues and would probably cost a lot of money. But I think we need to start somewhere, and I hope that the studies in my lab showing that it is possible. So just want to mention members of my lab working hard on these projects, and my collaborators and support from the funding agency. Thank you so much.
Okay, we have some time for some questions.
Thank you for the nice presentation. I have two quick questions. For the studies that you use microRNA with nanoparticles, so did you use just the active microRNA without nanoparticles in this study and to see the effect of this microRNA? This is my first question. just as a microRNA without nanoparticles as a control in the study?
Can I answer that question right
Yeah.
immediately? So I think there's a little confusion. So we didn't use nanoparticles conjugated to microRNA. What we did use was nanoparticles conjugated to oligonucleotide that targets microRNA 10B and inhibits it. So if we just take an oligonucleotide by itself, it's an RNA molecule. It's going to be degraded in serum in 15 minutes. There's going to be no effect.
Okay.
But nanoparticles were conjugated to single-stranded oligonucleotide. And that oligonucleotide binds to microRNA 10B and inhibits it.
Clear. The second quick question, could you ever determine how much of this arrive in the tumor of what you inject?
How much of nanoparticles?
Of the active?
Active, yes, we did do biodistribution. You know what? I don't remember the number. It's in the paper.
Okay.
I'm sorry. I can send you the paper. I don't remember the number.
It's okay. That's okay. Thank you.
So, any other questions? No, I have a question. So, especially with the siRNA delivery, right? There's only a percentage of the tumor cells that you'll actually be able to deliver your siRNA to, given the nature of nanoparticle delivery, which means that all the other tumor cells you don't silence anything. Like, what is the impact? Or like, what kind of numbers do I have to think of? Is it like 75% you will be able to silence, or is it only 25%, or?
Yeah. So we performed histology after the injection of these nanoparticles, and we showed that a very significant number of tumor cells is taking up those nanoparticles. I cannot tell you exactly quantitatively how many cells out of the tumor take these nanoparticles, but the thing is that, of course, all these treatments, they consist of repeating injections. It's not just a single injection and then you're done. So it's repeating injection, and in case of tumor, you actually destroy tumor cells, and you make it easier to deliver it to other cells that are not affected yet. So in our experience, after we're done with the treatment, not only we see the uptake of the tumor cells, but we also see massive apoptosis and massive necrosis, which tells us that those cells take up the nanoparticle with siRNA, and siRNA is acting up because the cells are dying.
All right. Thank you very much.